Okay, fine. So, hi everyone. Hi dear students. Hope I'm audible to all of you. So, guys, first of all, sorry for the break in the sequence of video that we were doing because of some, technical glitches. And today we will be talking about a very important topic, that is the glycogen, right? Glycogen is one of the very important topic, and for your first year exams, this topic will be in, most probably it will be in your long questions, right? Apart from MCQ, maybe there, MCQ may be there. But let's see what are the type of questions that you can get from this topic. Glycogen is little big topic from the carbohydrate metabolism you might have seen. Okay, so don't leave it. So what are the things that you should cover in glycogen metabolism? It is, first thing is like how the glycogen is synthesized, how it is degraded, right? Second thing, how it is regulated. So regulation of glycogen metabolism is very important. And fourth, that is glycogen storage disorders. So these four things that you should know grossly, right? So grossly we should know these four things. But apart from that, there can be questions which are like MCQs, reasoning based question, clinical cases based question. Okay, so like that. So that we will see definitely today, right? Okay. So see, like in the long question can be like this, like, describe the glycogenolysis, for example, with the steps and its regulation. So they are just asking glycogenolysis, that is glycogen degradation. So stick to that question, don't write the glycogen synthesis unnecessarily. They are asking only the question on glycogen degradation. Okay, so be, specific whenever you're writing your answers. And then questions can be reasoning based questions like this. Let's say why hypoglycemia is more severe in hepatic glycogen storage disorders, but less severe in the muscle glycogen storage disorders. McArdle's is a muscle glycogen storage disorder, but von Gierke's is affecting only liver, right? So like that. So such questions. Then there can be clinical questions like this, which can be part of your MCQs. Okay, that we'll discuss today. And then they can give you the clinical case, and then they can ask you the questions like this, like what is the most likely diagnosis? What is the name of the enzyme deficient? Okay, explain the biochemical basis of hypoglycemia in this patient. What is the inheritance pattern? And how does this disorder differ from McArdle's disease, etc., etc. Okay, so they can make such questions, not this much, like there are 6 questions here, but at least you can get 3 to 4 questions like that in the question that is part of the glycogen storage resource. Correct, guys? Okay, so let me quickly revise the glycogen metabolism. Right, guys? So, guys, what is glycogen, first of all? Okay, what is the glycogen? Just a second. So let's talk about what is glycogen. So this glycogen is a polymer of the glucose, right? So we all know it's a polymer of glucose. Okay, so how many glucoses are present when we talk about polymers? More than 10 glucose, right? So what is the definition of a polysaccharide? Okay, so we can use the word polysaccharide. Okay, it is a polysaccharide. Okay, and it's a polysaccharide of the glucose, right? Glucose when more than or equals to 10 glucose is present. That is called as a polysaccharide. So you got the point, guys? Now, the question is like why, you know, we are making glycogen. So obviously we are making glycogen so that our body, especially the liver and muscles, they have a storage form of carbohydrates, right? For the emergency supply. right? Like when I'm not eating for the next 12 hours while I'm sleeping, let's say. So what is the supply to my body? What is the supply to my heart, my RBCs, my brain, which constantly needs the energy supply, right? Okay, definitely not heart. Why I'm saying not heart? Because heart is dependent mostly on fatty acids, right? So polysaccharides. Now the question is, that what is the structure of the glycogen? This glycogen is a structure which is having multiple branches. So suppose this circle is a glucose molecule. So we have multiple glucose molecules like this, and then there is a branch. Okay, there is a branch point. So structure will be like this. I mean, like, we don't know where does it start, where does it end. Just to understand, like, this will be highly uh, branch structure. Okay, like this, like a tree. Okay, branches like a tree. So like this. But just to understand, guys, this is called as alpha 1,4 bond. Okay, alpha 1,4 bond. And this is called as alpha 1,6 bond. Fine. So these are the basic bonds that you should know. So can you see here that the bond alpha 1,6 is the bond which is present at branch points, and 1,4 is present at linear chain. Okay, so when I say the enzyme which is called as a debranching enzyme, so it should be called as alpha 1,6 glucosidase. What the point? Alpha 1,6 glucosidase. And if I call it alpha 1,4 bond, right, then this is alpha 1,4 glucosidase. Basically, guys, the phosphorylase enzyme of glycogenolysis breaks alpha 1,4 bonds, not 1,6. But the debranching breaks 1,6. You all know this? If you don't know, it's fine. We are just quickly revising it in a very simple manner, right? So guys, this is called as the structure of glycogen that you should understand, right? Okay, now question arises that why we are storing glycogen. Suppose this is a reasoning based question you get in your exams. But why human beings need to store glucose as glycogen? Why can't I directly store the glucose? Why this, hard work has to be done by the liver or the muscles or the kidney, right? So the reason being is that why I'm storing glycogen is, no, it's not like, it is compact form of storage. No, the answer will be, the best answer will be what? That glycogen is non-osmotic. Okay, glycogen is non-osmotic in the nature. Right? It is non-osmotic in the nature. So that is why glycogen is stored, right? But guys, glucose is osmotic in nature. So I can't store glucose in my liver cells, because if I store glucose in my liver cell, it will, what it will do? It will basically take the water inside to the liver cells, and that I don't want. Correct? So that is a glycogen, why we store it. Now, guys, first thing is glycogen synthesis and its regulation. Second thing is glycogen degradation and its regulation. And fourth thing is glycogen storage diseases. Right? Okay. So when we talk about the glycogen, right, synthesis. Okay, where we store glycogen, guys? Glycogen storage occurs mostly in liver, right? Liver has the most dense form because liver is a small organ, hmm, and liver is able to store 100 grams. It's 1.5 kg organ and storing how much glycogen? 100 grams. Muscles, they make like almost 30-40% of your body, but they are storing just 300 grams, right? So that is less density. So which is more dense storage? Obviously, the liver has a more dense storage. Okay, fine. And guys, liver maintains the blood glucose. No, what maintains the blood glucose? Liver, not the muscles, right? So that is why diseases which are liver glycogen storage disorders, they present with hypoglycemia, not the muscle glycogen storage disorders. Suppose like McArdle's disease, muscle glycogen storage disorder, but muscle glycogen is not source of blood glucose. Why? That can be again a question that we'll see. Okay, so let's talk about glycogen synthesis. So guys, this occurs in fat state, means I will take my meals. Okay, and what I need to synthesize it, I need number one, I need a primer protein. Guys, what is a primer? Primer, like in DNA polymerase. When we start PCR, we need a primer, no? What is the primary short fragment of DNA which help initiation of DNA replication, right? That is called as a primer because it is providing 3' OH group. So why we need a primer here? Because this will provide the attachment site for the glucose. So it's a primer protein actually, right? Primer protein. What's the name of the primer protein? We have glycogenin, right? So that's called as a glycogen. What else we need, guys? We need UDP glucose. So what is UDP glucose? So when I consume glucose in my diet, it first converts into glucose 6-phosphate, then it converts into glucose 1-phosphate, by a mutase enzyme. This is a mutase enzyme. What is enzyme here? Obviously hexokinase or glucokinase in the liver. So glucose 1-phosphate, and then I will be adding UTP, uridine triphosphate, and this will give me a highly active form of the glucose, and that is called as, what do you call this? This is called as the UDP glucose. Correct, guys? So that's called as UDP glucose. Fine. Okay. Fine. So that's how the UDP glucose is being made. Now the number 3. What is the third thing I need? I need the rate limiting enzyme that's called as glycogen synthase. So always be ready with whenever we start a synthetic pathway. Now you should be able to know that what I need to synthesize it. Any topic, let's take, for example, let's say protein synthesis you're doing, translation. So in translation, when you start, what do you need? You need a mRNA which is already modified. You need a tRNA which is attached with amino acid or charged amino acid, right? Charged with amino acid. You need a ribosome, that is ATS, and you need one more thing, that is the messenger RNA. So messenger RNA, tRNA, ribosomes. That's it. Three things that you need for translation process to begin with. So always remember like this. So when we are synthesizing, like when you're synthesizing fat or cholesterol, you need lots of acetyl-CoA, right? So we'll start with acetyl-CoA. Ketones also you need acetyl-CoA. Okay, so that's how we can remember. So always try to remember, okay, to synthesize this, what I need. Glycogen synthase, guys, this is the rate limiting enzyme. Okay, number 4th, what I need. What else do I need? So I need a branching enzyme. Okay, branching enzyme is also required. So which makes the branches. So that's it, guys. We will be able to make the glycogen like this. Okay, so when I have a primer protein, so suppose this is a primer protein. On this primer protein, we will keep on adding, glucose. Suppose this was a glycogen present before I took my breakfast. Residual glycogen present in my liver. So that was very less now. It is very less because it was present in the morning. In the whole night, I was using glycogen. So this is very less amount of glycogen being present in the uh, liver. So these are the, glucose is already attached. Now suppose if I change the color and take this blue. So now I'm adding more new glucoses, right? I'm adding glucose in 1,4. Okay, like this. So I will keep on adding, and guys, this addition requires glycogen synthase and UDP glucose. So I took UDP glucose and I took glycogen synthase. I have already taken primer protein, and now I will make the branch. Okay, so I can just break it. Suppose if I break these 3 glucoses and I just put them here, 1, 2, 3. I cut it from here, no? And I put here. So I made a branch. That's how I got a branched glycogen. That's it, very simple. Okay, so you can show that, initially I had a primer protein. On the primer protein, I had initially some small amount of glycogen. Then I add some more glycogen. Then I break it and attach here like this to make the branch using the branching enzyme. Clear? So that's it. That's how the glycogen is being synthesized. Okay, so guys, what is the rate limiting enzyme here? The rate limiting enzyme is obviously the glycogen synthase. First thing to remember. Second thing, guys, we should remember Guys, glycogen synthase is present in two forms. A, that is active form, and B, that is inactive form. So glycogen synthase A, glycogen synthase B. Now, if we talk about the regulation, okay, so guys, glycogen synthase, it is, allosteric. If I talk about allosteric regulation first, allosteric means the molecules. Hormonal is different regulation using insulin glucagon hormones, and it is allosteric. So if I take allosteric like glucose 6-phosphate, so guys, this is a positive allosteric activator. So means it will convert B to the A, right, form. So it is a positive allosteric activator. Now, second is glucose, right? It is the inhibitor, especially in liver, right? So in the liver cells, right? This is the inhibition of the glycogen synthase. This is the activation of glycogen synthase. And also, guys, in the muscles, ATP. If I take ATP, that is again the inhibition of glycogen synthase. That is in muscle. Okay. So now you see in the liver, free glucose, if it is present, it will inhibit the glycogen synthase. Okay. So if free glucose is present, it will inhibit the glycogen synthase. And in the muscles, if ATP is sufficient, that will inhibit the glycogen synthase. Correct, guys? So that is the simplest regulation that you can mention in the exams, right? And obviously, guys, glycogen synthesis will take place in presence of which hormone? Tell me, which hormone will activate glycogen synthase? Synthetic phase. Obviously, whenever I want to synthesize something, it should be fat state, right? When I'm breaking something, it should be the fasting state. And we know the two hormones of fat and fasting. Fat is insulin, and fasting is glucagon, right? So whenever we are in fat state, guys, fat state, there is increase in the level of insulin. And insulin will activate glycogen synthase enzyme. That is glycogen synthesis, increase in glycogen synthesis after the meals. That is what happens, right? So insulin is the enzyme. But guys, in the fasting state, when we fast, so in the fasting state, there is increase in the levels of glucagon, as we all know. And guys, what is the secondary messenger of glucagon? As we all know, the cyclic AMP. And this cyclic AMP will, activate the, kinases enzyme, which will phosphorylate and inactivate the glycogen synthase, right? So this will activate some kinase enzymes, and this kinases will phosphorylate, okay? Phosphorylate and inactivate, okay? Inactivate the glycogen synthesis, or glycogen synthase enzyme. So that means what? That means that glycogen synthase is active in dephosphorylated state in the presence of insulin, right? Guys, what insulin does? Let me write here. Insulin, let me take this red color. So guys, insulin, if you remember, insulin activate one enzyme, and that is called as protein phosphatase. Okay, protein phosphatase. Do you know these basic things? That, let's say the glucagon activates the, adenylate cyclase and makes the cyclic AMP, and then activates the protein kinases. So what does insulin do? Insulin do two things. First, it activates an enzyme called as protein phosphatase. So guys, this will dephosphorylate, you know, dephosphorylate the glycogen synthase enzyme. That is the active form, right? This is my A form, that is active form. Okay. And insulin do one more thing, guys, and that is activation of phosphodiesterase. Okay, activation of phosphodiesterase. So phosphodiesterase is an enzyme that breaks the cyclic AMP and inhibits the action of glucagon or terminates the action of glucagon. You know this basics, right? Okay, I can write here again. Insulin do one more thing. So if it is insulin, insulin will activate an enzyme that's called as phosphodiesterase, which will degrade the cyclic AMP. Okay, so this will degrade the cyclic AMP. Got it? So that's your, the basic regulation of, the glycogen metabolism, glycogen synthesis, right? Okay. Now, guys, just to understand better, I am writing two diseases here, right? So if I talk about two disorders here, what are the two disorders? Number one is glycogen storage disease, GSD 0. Right, what is 0? So guys, this is the deficiency, right? Deficiency of which enzyme? That is called as the glycogen synthase. Okay, first step. So what will happen in this disease? So in this disease, there will be nil or almost zero amount of, glycogen in the liver or in the muscles, right? So that means that this patient cannot store glycogen, cannot store now. So that means liver has no glycogen because cannot make the glycogen. So what will happen? So whenever this patient consumes a food, so in the fat state, there is increase in the glucose levels, that is hyperglycemia, right? Or increased glucose in the urine, that is glucoseuria, can happen to this patient. And in the fasting, what will happen to this patient? Because this patient has no glycogen in the liver, what will happen? There will be obviously what? Hypoglycemia, isn't it? Because there is no storage. So severe hypoglycemia will happen. So see again, this patient has no glycogen synthase. So when the patient takes the food, what will happen? The blood sugar rises, hyperglycemia. This sugar may reach beyond 180 mg per dl, that is a renal threshold, and may appear in the urine. So that is glucoseuria. In the fasting, this will cause hypoglycemia. Why? Because there is no ability of the liver now to break the glycogen because there is no glycogen. Simple, right? So that is GSD 0. There is no name of GSD 0. Now it's not type 2. I'm talking about now GSD fourth, right? We are talking about synthesis now, that's why. So this disease is called as the Anderson's disease. Okay, so what do you call this? Anderson's disease. So what is happening in the Anderson's disease, guys? There is a deficiency of the branching enzyme. Okay, branching enzyme is missing here. So deficiency of the branching enzyme. So guys, what happens when the branching enzyme is missing? So glycogen will not branch. Okay. Now first remember one thing that the branching makes, okay, branching more is the solubility. So branching makes the glycogen soluble structure. Okay, soluble. So branching makes the glycogen soluble structure. Okay. So that is your one thing to remember. So if there is no branching enzyme, so there is deficiency of branching enzyme means less soluble glycogen will present. And because of this, what will happen? Is glycogen precipitate. Okay. So glycogen will precipitate in the liver, and as this will cause liver injury. Okay, cirrhosis, you can say liver cirrhosis. That is one of the type of liver damage, scarring, fibrosis, and this will cause, liver failure and death. So as this patient's liver will fail, if the glycogen is like this, so it is not making any contact with water now, there is no branching. So that means it is less soluble, right? There are branching, but very less. Guys, remember glycogen branched every 10 glucose. But in this patient, the glycogen may branch at 50, 30, 20, whatever number of glucoses, but not every 10 or 20, 12 glucoses, right? 10 to 12 glucoses. So that's why it is insoluble. And that's why it is causing liver failure to this patient. Correct? So that is called as Anderson's diseases. Okay, now guys, this type of glycogen, which is less soluble glycogen, guys, this is also called as amylopectin. So I can say look like. So what is amylopectin? It's a type of starch. Starch is also a polysaccharide of the glucose, but starch is less soluble. And this is more soluble, that is glycogen. Just let's take this. Suppose this is glycogen branching very frequently. Okay, and suppose this is amylopectin branching very infrequently. So this is amylopectin, this is glycogen. So which is more soluble? Glycogen. More branching, more contact with water, right? Okay. So hope you understood. So these are the two disorders which are part of glycogen synthesis mechanism, glycogenesis. Okay, glycogenesis. Now let's talk about guys glycogenolysis. Glycogenolysis. So guys, this is happening in the fasting state. So why I have to break the glycogen in the fasting state? Guys, because to maintain blood sugar. So this can be again a question, guys, maintenance of blood glucose. Okay, so maintenance of blood glucose is a very important question that you can get as a short question. In that case, you have to mark everything about insulin, glucagon, and glycogen and gluconeogenesis. So what are the two metabolic processes directly involved in maintenance of blood sugar? Glycogenolysis, gluconeogenesis. So these are the two important processes which are very important for maintenance of the blood glucose. So let's take some basic points. First 12 hours, if you don't eat food, huh, then which maintains the blood sugar? That is liver glycogen. So major source of blood glucose is liver glycogen. Again, listen what I'm writing. How to maintain blood sugar in fasting state? First 12 hours, liver glycogen. Then there is around 13 to let's say 17 hours. If I say 13 to 17 hours, then glycogen plus glycogenolysis plus gluconeogenesis. So first is gluconeogenesis and glycogenolysis, both are required now. Now, if it is more than 18 hours, suppose if more than 18 hours you're not eating anything, right? What will happen, guys? This is very important again. So the liver glycogen, okay, liver glycogen is depleted. So if you're not eating more than 18 hours, liver glycogen finished. And obviously now gluconeogenesis is the only, right, only source of glucose, blood glucose. So guys, that is the reason, like, why we should not fast more than 18 hours. Why? Because gluconeogenesis will start majorly and it will break our muscle amino acids. So we don't want to lose muscles now. We don't want to lose muscles. I want to lose fats, not muscles, right? So we should not fast for longer hours because that is costing our amino acids from the muscles, right? Making us weak. Huh? So there are better ways of burning fat, not just fasting. Only fasting, I meant to say. So if you're fasting, if you ask someone like, what you should fast for 12 hours and you will burn more fat. Yes, you're right. But then after 12 hours, you should eat good protein diet so that your muscles don't suffer. So that protein will now be used at least for gluconeogenesis. The gluconeogenesis process will not use your own muscles. Take it from outside. Now I will take the protein supplement from outside to make glucose in case of fasting. In case of prolonged fasting, I mean to say, like to repair my muscles or to maintain the amino acid pool. Okay, guys, so that is how the blood sugar levels are maintained for first 12 hours. Fine. Now, guys, glycogenolysis. What is the rate limiting enzyme we have? So tell me the name. Glycogen phosphorylase. So that's the name. Okay, glycogen phosphorylase. Now, guys, this glycogen phosphorylase is the rate limiting enzyme. So which vitamin it is dependent on? Just a second, please. So it is dependent on which vitamin? It is dependent on vitamin B6. So it is dependent on vitamin B6, pyridoxal phosphate. Vitamins will be very important topics for your exams. So this is a rate limiting step and glycogen phosphorylase. So guys, what does this enzyme do? This enzyme only break alpha 1,4 bonds. So guys, remember and write. You can mention this in your exams that it doesn't break alpha 1,6. You know, and highlight it to make more impression, right? So breaks the alpha 1,4 bond, and guys, this will release the glucose 1-phosphate as the main product. So not the free glucose. See, glucose 1-phosphate. Let's again understand. Suppose this is my, you know, glucose, right? So glycogen, sorry, right? So it was having, let's say, multiple more branches. So I mean, like, what does the enzyme do? This, just a second. So what does this enzyme do? Phosphorylase, it will break glucose one at a time. Suppose this is the alpha 1,4 bond now. It will break it, cut. So cut this glucose out. Okay. And this glucose will be taken as glucose 1-phosphate. So it will cut again, take this glucose as glucose 1-phosphate like that. But guys, it will leave, so what is the major thing that I'll get? Glucose 1-phosphate. But now I also get 1, 2, 3, 4. 4 glucose still left at the branch point. This phosphorylase cannot break the last 4 glucose. Suppose it had 12 glucose, the chain of 12 glucoses. 8 I will take with the help of phosphorylase, but still 4 remaining. Na? So this will be called as limit dextrin. What do you call this? So guys, what are the products of glycogen phosphorylase reaction? Again, glucose 1-phosphate is a major product, plus remaining glycogen is called as limit dextrin. On this limit dextrin, I told you already, this is the bond which is alpha 1,6. So what we will be doing is that first I will remove the 3 glucoses, and the name of enzyme which do this is called as debranching enzyme. Now remember, debranching enzyme has two functions. First, it takes the 3 glucose out. Suppose, I have taken these 3 glucoses out now. So I'm left with only this. I'm left with only this alpha 1,6 bond. So now debranching enzyme, actually we call this as alpha 1,6 glucosidase. Okay. So that's the other name of debranching enzyme. Please remember it. Alpha 1,6 glucosidase. So guys, now I will be getting free glucose, right? One from each branch, one free glucose. I have taken only the figure which has shown only one branch now. I'm not taking a tree-like structure here. So one free glucose I got. So again, understand. First, I took phosphorylase. Huh? Phosphorylase cuts the glucoses, removes every glucose as glucose 1-phosphate, leaving the 4 glucose at the branch. These 4 glucoses are removed by debranching enzyme. First, it removes the 3 glucoses by breaking alpha 1,4 bond. But now I have to break alpha 1,6 bond. So that will be broken by the second activity of debranching enzyme called as alpha 1,6 glucosidase. So guys, still I have what is the major product? Not the free glucose. What is the major product? Glucose 1-phosphate. So what to do next? So I'll be taking glucose 1-phosphate, then I'll convert this into again glucose 6-phosphate. And guys, in the endoplasmic reticulum, so in the enzyme section, when we read one topic that is called as marker enzymes. So endoplasmic reticulum has an enzyme that's called as glucose 6 phosphatase. Okay, this is called as glucose 6 phosphatase. So guys, this glucose 6 phosphatase will make the glucose, free glucose in the liver, and this will appear in the blood as a blood glucose. And obviously from the blood, it will go to my main cells like brain and RBC, which cannot survive at all without the glucose. So brain and RBC will use it. Right, guys? So that's how, it will happen. So brain and RBC will be able to use it. So that is our next step. Clear? So this is how the glycogenolysis occurs, guys. But guys, there is a very important question here. Like, why do muscles cannot make free glucose in sufficient quantity? Yeah, at all, right? So remember, muscles, muscle cells, they lack glucose 6 phosphatase enzyme, right? So, guys, what is the important thing about glucose 6 phosphatase again is it is also required in gluconeogenesis and also required in glycogenolysis. So, two pathways which are simultaneously, I mean, like, no, not simultaneously, but two pathways which have a common enzyme. Okay, so what is a common enzyme? Right? The common enzyme of the two pathway is glucose 6 phosphatase. Understood? Understood? So deficiency of this enzyme will affect two pathways, only two pathways in our body which maintains a blood sugar. So deficiency of this enzyme will lead to the most severe hypoglycemia because you are not able to do the gluconeogenesis also. You're not able to do glycogen breakdown also. So that is like one of the serious condition, von Gierke's disease. Though a rare disease, but yes, that is a serious condition, right? So guys, muscles lack glucose 6 phosphatase, and that is the reason. So, not a source of blood glucose. So, what does that do then? Huh? So, they use, you know, glycogen like this. Glycogen convert into glucose 6 phosphate is the main product. So what is the major product of glycogenolysis in muscles? Glucose 6 phosphate. This glucose 6 phosphate then, guys, goes into the glycolysis, and this will produces some ATP for the muscles, right? So ATPs will be produced. So guys, when you go for exercise, for your run, what do you use? You use some muscle glycogen as a source of energy, right? So we break the muscle glycogen to G6P, and then this makes a ATP, correct? So that's how the glycogen, in the muscles have two questions again. Why muscle glycogen is not a source of blood glucose, I explained. And what is the major source of, what is the use of muscle glycogen then? It uses for its own benefits, like to make the ATPs during exercise, right? So that is exercise basically during the exercise. Guys, how much ATPs it will make? It will make one extra ATP now. See, I am starting glycolysis not with glucose. I am starting glycolysis with G6P. Hmm? So, muscles don't use hexokinase step in above case, matlab glycogen case, right? So, one extra ATP made. So like if it is making 7, I'll be getting 8. If it is anaerobic glycolysis during my exercise, I'll be making 3 instead of 2. Normally in anaerobic glycolysis, how many ATPs are made? 2. Now I'll be making, if I use glycogen, 3. Correct? So that's why like muscles will make 3 ATPs during exercises. One extra ATP in exercise. Right? So I'll be getting one extra. Understood everyone? That I'm not using hexokinase because I'm starting with G6P. My glycolysis doesn't start like this. It's not glucose to glucose 6 phosphate. I'm not using this step. I'm starting directly from here because this is made from glycogen. So glycogen made this, that's why. Okay, so guys, these are some more reasoning question I explained to you. Okay, if I talk about regulation, right? So regulation, guys, remember in the muscles, guys, calcium. So remember calcium. So when I start exercises, and when I start exercises, ATP converts into AMP. I had ATP, now I start exercise and AMP is made. So I'm removing ATP from here. So this calcium and ATP, both of them activate the phosphorylase. Okay, phosphorylase enzyme directly or indirectly. See guys, this can be directly or indirectly. Like calcium can do indirect way. You know how calcium activates enzyme phosphorylase kinase first. Phosphorylase kinase and activate the glycogen phosphorylase, which was inactive to the active form. So that is some extra inputs that you can add to your exams also, no problem. But end of the day, you should be able to remember, ki, okay, what is the muscle, calcium and AMP, why? Because as soon as I start exercising, calcium will be released and ATP will be broken to AMP, and that's the need of more energy. What is AMP? Signal of low energy. So I need more energy, right? So that is in the muscles. Now, guys, in the liver, if I talk about, okay. But guys, remember one thing. Muscles have no glucagon receptors. So generally, glucagon, we don't talk in muscles. Glucagon increase blood sugar by activating the glycogen phosphorylase in liver, not in muscles. Right? Muscles don't have glucagon receptors. Probably this is the way of nature saying that only liver will maintain the blood sugar, not the muscles, because muscles also don't have glucose 6-phosphatase. So why to just add a glucagon receptors there? Understood now? Because glucose 6-phosphatase the last time is already missing, so they will never make any glucose muscles, I meant to say. Now in the liver, guys, again the same thing. Whenever we fast, there is increase in glucagon, right? So there is increase in the secondary messenger cyclic AMP, which activates the enzyme that's called as protein kinase A. Guys, this protein kinase A then activates one more enzyme that's called as one more kinase, phosphorylase kinase. And this phosphorylase kinase will convert, guys, the B form of glycogen phosphorylase to the A form, that is active form of glycogen phosphorylase. This is active. And this will cause glycogenolysis. A. So now they will be increased glycogen breakdown. So see guys, how does glucagon works? Cyclic AMP, protein kinase A, phosphorylase kinase is one extra step here. Right? Remember, phosphorylase kinase is actually a kinase of phosphorylase enzyme. So this enzyme is active in the phosphorylated state. B had no phosphorus attached. This is like, covalent modification of enzyme. No? I have now attached a phosphate here, PO4 phosphate. So this is the active form, right? So this is glycogen phosphorylase B converts into A. That's it, guys. This is all about the regulation part. Regulation can be a short answer question in your exams also, right? Okay, fine. So talking about, guys, the questions that we took in the beginning. So let's take the question here. So a woman's 6-month-old infant is brought to the pediatrician due to frequent episodes of hypoglycemia, severe hypoglycemia, right? Especially when fasting. On examination, the child has swollen abdomen, hepatomegaly. Guys, more and more glycogen stored in the liver, hepatomegaly. Lab investigation shows high lactate, hyperuricemia, hyperlipidemia, etc. Liver biopsy. Now what happened? So, liver biopsy reveals excessive glycogen with a normal structure. Okay, so question is, what is most likely diagnosis? So let me tell you this question. Let's say how we will write. So I will write the my most likely diagnosis is von Gierke's disease that I will discuss now. Von Gierke's disease. Huh? So that is my likely diagnosis. Something has happened. Fine. So that is our diagnosis is von Gierke's disease. So this question is done. Which enzyme is deficient in this condition? Glucose 6-phosphatase, right? Remember, like von Gierke's, when I say von Gierke's, glucose 6-phosphatase is missing. Okay, then explain the biochemical basis of hypoglycemia and lactic acidosis in this patient. So, and also hyperuricemia and also the hyperlipidemia. So let me explain this. And what is the inheritance pattern? That's not important, right? How does this disorder differs from McArdle's disease? You know, guys, this disorder doesn't affect muscles. Why? Because glucose 6-phosphatase is already absent in muscles. So this disease affects only liver, not the muscles. And McArdle's affects only muscles. What is the treatment? Never allow fasting. You know, in this patient, you have to write that don't allow fasting to this patient, because when this patient fasts, glycogen cannot be broken. Okay, last step is missing now, glucose 6-phosphatase. So obviously this patient is unable to break the glycogen. So don't allow the patient to fast because he is unable to maintain the blood sugar level. So keep giving baby food every 2 to 3 hours, normal like that. Not throughout the life, the patient has to do this. Okay, so it will be like this way, guys. Fine. So let me explain some of the GSDs. So glycogen storage disorders. The first is von Gierke's disease. Now see guys, in the von Gierke's disease, glucose 6-phosphatase is missing. The first important disease is von Gierke's disease. So glucose 6-phosphatase is missing. So guys, what will happen because of this? So there will be increase in glucose 6-phosphate. Fine. And this glucose 6-phosphate will enter into HMP shunt. And there it will be producing riboses like ribose 5-phosphate. This will make purines and this will make uric acid. So guys, that is why this patient has hyperuricemia in our MCQ. Got it? Hyperuricemia in our patient. Now, because glucose 6-phosphatase is missing, that means decrease in glycogenolysis, decrease in gluconeogenesis. I told you already that this is the common enzyme of both the pathways. So guys, this will lead to severe fasting hypoglycemia. So that is why this patient should never fast. So what is the treatment strategy? You know, what is the management? Feed every 2 to 3 hours, right? Like that. Then glucose 6-phosphatase is missing. So increase in glucose 6-phosphate. Now with this increase in glucose 6-phosphate, I told you already, activate the rate limiting enzyme glycogen synthase in the liver. And guys, because of this, increased glycogen synthesis. So glycogen synthesis will increase, and as this will cause massive, bigger, very big, massive hepatomegaly, swollen abdomen as a question says. So massive hepatomegaly will be there. So hope you understood guys these 3 points. Now, again our question, why there is, let's say, lactic acidosis. So guys, because there is increase in glucose 6-phosphate. So this glucose 6-phosphate will go to glycolysis, because it is unable to convert into glucose in the liver. So now it will be used by the pathway of glycolysis. Now in the glycolysis, definitely I get pyruvate, but excess pyruvate will not be directly used for various purposes. So this pyruvate can be used for 2 purposes. I will convert this into acetyl CoA and will make fats out of it. So guys, that's why hyperlipidemia, increased TG levels will be there. And I can make pyruvate to lactic acid. So guys, lactic acid doses. Now you can ask me that why it is, lactic acid is being accumulated, because lactic acid should be utilized by the gluconeogenesis pathway. Because gluconeogenesis, lactate is one of the important source of gluconeogenesis now. Then why the patient is unable to use it? Because gluconeogenesis is also decreased now. Glucose 6-phosphatase is missing. So gluconeogenesis also decreased. So that's why lactic acidosis occurs. So hope you understood why there is hyperlipidemia, why there is lactic acidosis, etc. etc. Fine. So that is our, how we can do the long question. Fine. Now, what are the other diseases, guys? Let me just summarize it. So, what is a type 2 disease? It is called as Pompe's disease. Guys, what is the specialty of this disease is that this is the disease where lysosomal alpha-1,4 glucosidase, right, enzyme is missing. Or what is the other name of this enzyme? Acid maltase. So acid maltase is missing. So these are the two names of the same enzyme. They may be missing in the Pompe's disease. Clear? So that is missing in the Pompe's disease. That is type 2 lysosomal storage, glycogen storage disease. Guys, this is the only glycogen storage disease which is also a lysosomal storage disease. Can you name me one more lysosomal storage disease that you have read? Like, for example, you might have read the Gaucher's name, then you might have read, the San Filippo's, then Tay-Sachs, etc., Niemann-Pick's. So these are also the lysosomal storage. Hurlers, Hunters. So they are all examples of lysosomal storage disorders. Correct? So this is the only GSD which is also lysosomal storage disease. What is the third? How will you identify third? Third is Cory's disease. And what is the name of enzyme missing here? ABCD, Anderson branching. So Cory's is D branching. So D branching enzyme, if it is missing, that is alpha-1,6 glucosidase. Okay, that is missing. So guys, what will accumulate? Limit dextrin cannot be broken now. So we also call this disease as limit dextrinosis. So guys, name two diseases where abnormal glycogen accumulates. So answer will be type 3. Limit dextrin type of glycogen accumulates. You know what is limit dextrin? This is glycogen with short branches. Understood? Short branch. So glycogen with a short branch. I mean like this. Branching is proper, but short branches. Why? Because they're not broken now. So I called the patient in the morning. I took the biopsy, and patient do not have in the morning such long branches. But what is left is this. So they should not left now, because if digestion was complete, I mean degradation of glycogen in the liver was complete, it should be the glycogen with normal structure should be visible. Whatever is left, not the short branches are left, because alpha-1,6 glucosidase is absent. So name two disease in which the abnormal glycogen is accumulated. Type 3, limit-dextrin accumulates. Type 4, amylopectin, unbranched type of glycogen is accumulated. Understood? We'll be able to do it now. So that is type 3. Type 4 I have already done. Guys, type 5 is called as McArdle's disease. Okay, McArdle's. So let's take the question first. We have a question also. So, so this question, MCQ, 20-year-old athlete reports muscle cramps, fatigue during intense workouts. His symptoms improve, he takes a break and eats something sweet. Lab shows myoglobinuria and hypoglycemia, right? And the hypoglycemia. And no hypoglycemia, sorry, no hypoglycemia, right? What is the most likely diagnosis? So if there is a question which says about the weakness of muscles, especially during the workouts, the answer is McArdle's disease. Okay, the best answer. And see, there is no hypoglycemia also. See, guys, hypoglycemia occurs mostly during liver diseases. This is a muscle disease now. So no hypoglycemia, right? This is a muscle disease. Clear? So McArdle's disease will be the answer. Such history you will get in McArdle's diseases. Okay, let's see this question. Why is hypoglycemia more severe in hepatic glycogen storage? I told you why. Because in the hepatic, only liver maintains blood sugar level. Na? So now you have to write this line that muscles lack glucose 6-phosphatase, so it's not a major source of blood glucose during fasting state. So this you have to write. This can be just a 2 mark or 1 mark question, 2 mark question for reasoning purpose. Okay, so guys, that's how we can explain the glycogen metabolism disorders. I have already explained, right? McArdle's, if I talk the last point, that is McArdle's disease. So what happens in McArdle's disease? There is a deficiency of muscle. See, Mac, M for muscle, McArdle's muscle phosphorylase. So muscle phosphorylase is missing. Glycogen phosphorylase. So this is a glycogen phosphorylase, obviously, rate limiting step. So that means if muscle had glycogen, it is never broken to glucose 6-phosphate. This is the deficiency here. And glucose 6-phosphate never converts to, I told you already that it never converts to glucose. Fine. So obviously we don't get glucose here, or we ideally goes to glycolysis. है ना, ये तो होता ही नहीं है. So glycolysis we have to do, and in the glycolysis what we make? We make the during exercise lactate. Anaerobic glycolysis, na? If it is exercise, it is anaerobic glycolysis. So forget this. Obviously, this enzyme is missing already. So guys, in this patient, you will not even find the lactate during the exercise. See, when you do exercise, even for small time, your blood lactate will rise. There will be no rise in lactate now because pathway is missing here. So I can write this line that no increase in blood lactate, after exercise. Right? And why this myoglobinuria? Because muscle breaks. When muscle is not getting energy, the muscle fibers may break. And this can release a myoglobin and can cause even the kidney injury. Okay, so or the urine has myoglobin. So such way. Right, guys? So as these are the 5 important disorders that we have discussed. Type 6 is Hirsch's disease, hepatic phosphorylase, though it should be a very serious condition, but patients are very less till now identified, and it was found to be very mild disease. The reasons are unknown. Ideally, it should be a very serious disease because it is affecting liver directly. So it should have serious hypoglycemias. Okay, fine guys. So that's all about. Hope you understood that what kind of questions you can see again that can be asked to you in the exams, right? Questions that can be asked to you in the exams. Reasoning type, short answer, MCQ, and the long answer question which may have clinical case, or the simple long answer question like this. Describe glycogenolysis, steps and regulation. So simple it is, and they will be, like this. Steps you will be getting, let's say, 5 marks, and regulation you're getting 5 marks. Okay, and they may ask you the diseases associated with glycogenolysis, 5 marks. So 5, 5, 5, that makes total 15, or maybe any distribution you can get. So guys, thank you so much. Okay, and I'll see you again. So keep revising. Yeah, done. It's finished. Okay, sir. I'm closing. Okay.